So, hi everyone, and welcome to today's webinar. My name is Paul Adepoja, and I'm the Community Manager for the ICFJ Global Health Crisis Reporting Forum. And today we have an interesting topic to discuss today. And it's a topic that has consistently been in the headlines for quite a couple of weeks now. And for individuals like our panelists today, it's part of what they do routinely. But for the rest of the world, people that are not familiar with viruses, with the genomics and how variants really work, it may be quite difficult to understand what variants are. So that's what we are deciding to focus on today. And the goal of today's webinar is to guide our journalists from different parts of the world on how to accurately and expand their reporting on the Delta variant. So I'm really glad to be joined by Peter today. He's joining us from the South African Bioinformatics Institute. Peter, how are you doing today?
I'm doing well, thanks, Paul. Thanks for inviting me.
Yeah, I'm really glad to be hearing from you today. And I really look forward to your presentation. And I would like our audience to know that the Delta variant is not a particular variant that is different from, that's only affecting one part of the world. It's gradually it has become a global phenomenon. So wherever you are, this topic is of particular interest, and I look forward to our interesting discussion. So if you have questions during the course of the presentation, please use the Q&A section on the Zoom platform. And if you are joining us on the Facebook, please put your questions under the live stream video that you are watching right now. So we'd like to start with Peter's presentation before the question and answer. So Peter, you can share your screen and go ahead with your presentation. Thank you.
Thank you. Yes, so let's talk about understanding Delta. So as Paul said, I am from the South African National Bioinformatics Institute. There in the background, you can see our offices at the University of the Western Cape in Cape Town, South Africa. I'm a bioinformatician there, and my focus is on pathogens and making bioinformatics more accessible in African public health, okay? So let's start off with a little bit of understanding SARS-CoV-2 as the background, okay? So you've heard about this virus, you've seen these pictures of the virus, and here we have a diagram of what the virus looks like. So it's this little bubble of membrane with spikes sticking out here. They look a little bit like tall mushrooms, and inside there's a thing called the nucleocapsid, okay? And that's wrapping up this thing called its genome, which is the stretch of RNA. Now, RNA, we can think of as a linear recipe like this, and it's broken into pieces. So we call them ORF, so ORF1A, ORF1B. Over here we have the spike, and these are the recipes for making the different parts of the virus. And these are other ORFs here at the end, okay? We're going to see that a little bit more in future slides. So some terms that we use in the field, we talk about bases. So the recipe that makes up the virus is this DNA, or in this case of this virus, RNA, which is made up out of these letters called bases, A, C, U, and G in the case of RNA. And the genome is about 30,000 of these things, which are the recipe that the virus uses. So it basically feeds the recipe to your cells once it gets into you, and then it uses that to make more viruses. And then to actually understand what this genome looks like, we use machines called sequencers. We use a technique called sequencing, where we basically get the virus as lots of little pieces. We put it together like a puzzle. And but we start with those swabs that they kind of put up your nose when they're doing a test. And then we eventually get to read the genome of the virus, okay? And when we're reading the genome, then what we do is we get it in all these letters of RNA, and all three of these letters makes up one letter of protein, so one so-called amino acid, but they can be changes. So as the virus is making copies of itself, you can think of it's working like a photocopier, but you know, photocopiers get those specks of dust and things like that, and you see some changes happening. But some of the combinations of letters can be synonyms, okay? So see this, here are the names of the different amino acids on the outside, but you see that a C, a U, and a U gives you this amino acid, and a C and a U and a G gives you the same amino acid. So sometimes there can be a change in the RNA, but it doesn't change the protein. But sometimes if you had a C and then the U changed to a CCU would change the protein that is being made. Okay, so not every change in the genome is leading to a change in the protein, but the proteins are the actual things that do the work of the virus. Okay, and how do we get variants? So when you get infected with this virus, some number of viruses get inside you, but you start off that we start off with a virus where it all looked the same. Small numbers of people were getting very much exactly the same virus. What I mean by exactly the same is if you take that all of the 30,000 letters of the genome, it would be exactly the same 30,000 letters between these viruses. But then over time, as the infection grows inside of you over a course of several days, then, as I said, this virus is not making perfect copies of itself. So you end up actually having a whole population of viruses with majority and minority variations of the virus inside you. Now, if you infect somebody else, most likely you will infect them with, again, the majority variation that's sitting inside of you, and then it will come and differentiate inside them. But what can also happen is that like a minority variation comes out. So now the person who's infected, the virus is looking slightly different. And you see these different colors of viruses over time as the virus spreads between the population, okay, of infected people. So it's infected people who are the engines of variation. So the more people be getting infected, the more different kind of colors of these kind of viruses you're going to get, the more different strains. So we call these descendant viruses, we call them lineages, okay? And we visualize them in something called a phylogenetic tree. So my slides will be available afterwards. I will put them on Twitter as well. And there's a nice guide out there from the US CDC on how to read a phylogenetic tree. You can go through that one. So a lineage is a path through this tree. We'll see a tree just now. And the lineages get these names called pango lineages. I'll talk about them just now. But if you took a look at all of the descendants that come from one group of lineages, then we call them a clade. And there's a project out there called NextStrain that has its own naming scheme called NextClade. So we have these two naming schemes, the pango lineages and the NextClade clades, okay? It's going to become simpler. So this is a picture of an early phylogenetic tree of the virus. This was the end of January 2021. So at that point, they'd only sequenced the virus in Thailand, here in China, and in some parts of the United States. Now, you can imagine that there was an original virus here at the start. Now, one of the interesting things is that if you look at the very earliest viruses, we actually already see two forms, okay? And now you see here, there's different paths through the tree. That's called a lineage, okay? But now look at this yellow bit. Everything that belongs here was descended from a virus that they had things in common. So somebody had an infection, they passed it to other people, and in this virus that is in this yellow clade is looking to this kind of teal colored one. Now, what is the difference? Down here at the bottom, we've got a picture of the genome. And here at the end, there is a region called ORF8, and something changed in ORF8 that meant that basically an S amino acid instead of an L amino acid was created. So everything that's kind of light blue here, at that position, which is position 84 in ORF8, you can read an L. But in the yellow ones, you can read an S, okay? Does that make a difference in terms of how the virus works? Well, at this point, no, the answer was it didn't. It's just like a random change. It's like changing the color of your car. It's not going to make it go faster or slower, okay? But we can use this to actually start talking about, well, okay, everybody with this yellow virus, that means they got infected by somebody else who had a kind of what I'm calling here the yellow variant virus. This was long before we had names like Delta and so on for the viruses, okay? So this variation lets us track the spread of the virus around the world. So then a little bit later on, this is March 2020, we had the emergence of what some people call DUG. okay? This is a different part of the virus that changed here. This is looking at the so-called spike protein, that kind of mushroom-like structure that I showed in the picture. And here at position 614, a D changed into a G. So D to G, that's why I'm calling it DUG, okay? And we see here that most of the original infections in China were the original form. They had a D there. But here in Europe, we start seeing this G, okay? And what happens as we move on? June of 2020, we see that DUG is taking over, right? Here in samples from South Africa, in the United States, in very much in Europe, and even in parts of Asia, we mostly are finding the G, not the D there. What this is showing us is that there seems to be something about the DUG variant that means it's doing better than the original variant, okay? Here's your original gangster virus, which had a D there, but the one with the G seems to spread faster and better, okay? And this was a topic of major debate, you know, did it really make a difference and so on and so forth, you know? But it seemed that over time, we kind of came to the conclusion that yes, DUG was doing better than other versions, okay? And when we look at like, does it make a difference? This is a picture of the spike, okay? And there, deep in the heart of the spike, that's where this DUG change happened. The D changed to a G there in the heart. When we sequenced the first sample that was sequenced in South Africa, then we saw this D to G change, but looking at the structure of the virus and where the change was happening, we thought it didn't make a big difference. This little red bit over here is the so-called receptor binding domain. That's where the virus sticks onto your cells on the so-called ACE2 receptor. This green bit over here is the so-called N-terminal domain, and we think that it's quite important in terms of how the body's immune system responds to the virus, okay? So we saw this change, and it wasn't in the receptor binding domain, it wasn't here in the N-terminal domain, so we didn't think DUG was such a big deal, but it seemed that it became a big deal over time, okay? But looking at this three-dimensional structure of the virus, instead of that like long line that we saw before, is how people can start thinking about, well, where does a change actually make a difference? Where does a mutation make a difference? So then we started getting this naming scheme. So we see all these numbers here on the right-hand side. These are the so-called Pango lineage designations, okay? So what this means is that all of these lineages are assigned by this group doing the pangolin software. The University of Edinburgh was doing this. Some actually PhD students were maintaining this list at the time. And that means that if we look at a B.1.1, we know that it descended from the original B.1, okay? So in our first wave in South Africa, this red line is the number of cases per day, and the bar graphs are the number of samples per month. So they're quite different scales, but they give you some idea of, you know, we were sequencing more during our waves and things like that. This was done by different labs in South Africa, okay? And we see that in the first wave, there was quite some diversity of different lineages of viruses circulating. So that means that it didn't look like any of these viruses or the different versions of virus were like beating each other. All of them were bad. All of them were spreading, okay? But then something started happening around September last year. And we see this blue section expanding and expanding. Now, if we look over here, it's B.1.351. Now, you know, you journalists didn't really like the fact that scientists had all these funny names like B.1.1.7 and B.1.351. Because they're not really like easy for people to understand. So we these days call B.1.351, we call it beta. And we see that the second wave, which was higher than our first wave, was driven by this beta, okay? And then it carried on, and we thought we saw the third wave starting to grow. And here in the samples, we still see here in May a lot of beta, but this orange thing starts growing. B.1.617.2, otherwise known as delta, okay? And you see this is drawn just as our third wave was really ramping up, and we start seeing that most of our samples are Delta samples. Now, it's important to realize that these numbers here, we've got maximum of about 2,000 samples in 1 month in South Africa, okay? And this is in the context of a situation where we're getting more than 10,000 infections every single day. So we're selecting a small part of the total collection of viruses out there. And we're hoping that we capture a representative part in the process, okay? But it's because sequencing, it requires equipment and resources and skills that's a lot more complicated than even testing. So we cannot keep up with every single virus out there that needs that infects somebody, okay? But we see worldwide that what I call the rise of the variant. So this earlier part, we have the SARS-CoV-2, we have COVID-19, and people like myself, bioinformaticians, looking at all of these genomic things, we're not really seen as I mean, we're seen as geeks that like are not that important in the bigger picture of things, okay? But here, December, we have two announcements from Public Health England in the UK and from the group at CRISP in South Africa, where they identify what, as I said, was called B.1.1.7. This is alpha in the new naming scheme, and B.1.351. And these have mutations in that core part of the spike that the part that's clicking onto the cell, okay? The N501Y, and then in the South African one, there's a mutation in something called E484K, 484 amino acid there in spike, okay? They're colloquially known as EEK, because E484K seems to allow the virus to escape immunity, even if you've been infected before, okay? So it's quite a concerning variant. So here in December, everybody starts talking about variants, and we see here this dark blue of beta starts dominating. Now, this isn't the reality of the pandemic. This is what we've been able to sample. So if you are not sequencing, you're not seeing these things. But where we can sequence, we start seeing this growing. And then we see alpha in the UK. The UK is doing the best sequencing in the world at the moment. So they're getting a lot of these alpha samples, and high proportions of the virus are these so-called VOCs, variants of concern. So that just alpha and beta by March are making up about 70% of all the viruses being sampled. But then something starts sneaking in, March, April, May, where we start seeing this terrible story of like a runaway pandemic in India. Now, when we looked at this thing, the first thoughts were, look, this is related to the easing of restrictions. This is related to political campaigning and religious festivals and various things that allow the virus to spread. But then this data starts coming in from Indian researchers that, look, a lot of what we're seeing is this thing called B.1.617, okay? And then that splits into different types, B.1.617.1, .2, and .3. It then gets through to the UK, where, as I said, in the UK, they've got very sophisticated sequencing happening, not in terms of the machines so much, they're using the same machines that we are using, but they have a really well-organized human machine of collecting samples and doing a lot of sequencing, okay? And Public Health England, you know, says, look, this thing is a variant under investigation, that VOI that we are looking at in April, and the WHO says, look, this thing is really worrying, and by May, it gets listed as a new variant of concern. And you see how this is not an African picture. This is a global picture. We see how Delta is just taking over. It's pushing all the other versions of the virus out of the way. It has what we call a selective advantage. We can see this on the tree. This is focusing on Africa. We see that here is the growth of beta from some samples. Here it grows until it's like there's a lot of beta in our samples. Here we have the other variants that are not variants of concern, but you see that over time there's less and less of them, okay? This over here is alpha, particularly in West Africa, we were seeing a lot of alpha being picked up. But then this teal colored thing, this is delta. So you see it gets detected later than these other clades, but it then starts taking over. And if we now look at the samples, we will see in the next slides, it is taking over the continent. So what do we know about Delta? Okay, now a lot of what we know about Delta is not based on research where it started. I mean, the people in the scientists working in India did some great work, but just in terms of the funding and the people and the ability to kind of come to conclusions, the research done in the UK was giving us more insight. So and now more recently, some of what I'm referring to here is actually from research done in China, where they've detected this thing in their kind of quarantine hotels, okay? So it is more transmissible. In other words, if you start off with some people infected with, say, the alpha version and other people infected with the delta version, over time, the people with a delta version will infect more people. It has what we call a higher R0, okay? And this seems to be because people have a higher viral load. In some recent research, they said that on average, people with this version of the virus, when they're at the peak of their infectiousness, which we must remember, people are actually at their peak infectiousness just as the sickness is kicking in. So you get infected and it takes some days for the virus to grow, and then you start like spreading the virus to other people, perhaps even before you fully notice that you're sick. This is one of the concerning things about this particular disease. So it's about 1,000 times higher in terms of the amount of virus that this person has inside them compared to the previous versions of the virus that we've seen, okay? So, and it might be rising faster. So again, in this report on the website called Virological, you can see by clicking on my slides later, they were looking at a quarantine hotel in China, and they were saying that basically from when it appears that they got infected to when they were getting sick, it was on average 4 days. Whereas with the previous versions of the virus, they were saying on average 6 days. So this is one of the reasons that this virus is like running like a fast burning fire through the people who can be infected. The data on whether the individual disease of an individual infected with Delta is more serious or not is still uncertain. It doesn't look like this necessarily causes much of a different disease. But you know, when so many people are getting sick so fast, your hospitals have a shortage of beds, and as we saw in India, very crucially, we have a shortage of medical oxygen. And even if the virus is no more deadly than the original version of the virus, then the fact that it's overwhelming your healthcare system can lead to an increase in deaths, okay? Meanwhile, there is some good news from those places that have lots of vaccines. UK and Canada, they've looked at the Pfizer, AstraZeneca, and Moderna. I know none of us in Africa have Moderna vaccines, and they all seem to retain effectiveness. They all are all still great vaccines against this version of the virus, if you can get them, okay? The problem that is, however, that, you know, we also have other vaccines in use on the African continent, but we don't have necessarily the research infrastructure to actually give us great data on how well some of the vaccines that we're using on the African continent, like Sinovac and the Sputnik vaccine, how well they're working. You know, we need strong regulatory bodies, we need strong scientific bodies to get that data, okay? And this is you know, there's a great book out there by my colleague Iruko Okeke called Divining Without Seeds, and this is where we're still sitting in 2021. Our eyes are still clouded, okay? Just an aside, you might hear about like waning antibodies, you know, less antibody response and things like that. They do this using, you might even see these graphs, a virus neutralization assay, okay? So they take somebody who might have been infected with SARS-CoV-2, or they maybe been vaccinated, they purify their blood, and they get the antibodies from there against a virus in a little lab petri dish, okay? And then they come up with answers. These answers should be taken with a grain of salt, okay? Because what's happening with antibodies in a little petri dish or in a test tube is not the same thing as what's happening to your immune system, because your immune system has T cells and B cells and it's got multiple layers of defense. So when people go on about like, look, this resistance or your immunity is less effective, and then they're only looking at antibody studies, realize that that is only a limited lab-based study and that you might want to wait for more thorough information. Okay, so that's all a bit about virus biology for a second. As I said, we've got a problem with eyes. This is a picture of the number of samples submitted to the GISAID database, which is one of the main databases where we share virus data from the African continent. You see South Africa at about 12,000 samples has by far done more than anybody else, okay? Then there's Kenya, Egypt, Ghana, Nigeria, Angola, etc. Okay. As some of these, for instance, Angola, the samples are collected there, but still sequenced in South Africa. So we've got a problem with not looking across the entire continent. Then has this improved over time? Somewhat. This is the date of submission of samples, and we see that here it grew steadily over 2020, and it has been growing, largely driven by growth in the South African sequencing efforts since the start of 2021. But it's still like we're talking about, maybe 6,000 samples a month from a continent that's having many, many, many more cases in that. And this is not evenly distributed, the surveillance across the continent. The other problem is this. This is how long it takes for a sample that was collected to end up in the databases, okay? So now the databases are not the only place where there's information. So for instance, when my colleagues in the DRC pick up a sample and they find out something really interesting, they can inform the Africa CDC or the World Health Organization's African and Regional Office very rapidly what the results are. And it might take some time before the Ministry of Health says, okay, it's fine for you to publish the data in a public database. So that means the information might be available before we see it in public, but still the time to see it in public is a useful metric. In most of the continent, the average time to see the data in public is over 50 days. So somewhere close to 2 months, okay? South Africa and to some extent Egypt are doing a little bit better, but even for us, the average time from collecting a sample from somebody's nose or whatever it is to getting the sequence data out there for public consumption is on the order of 1 month, okay? So that means that we are constantly looking into the past. And we see that here, that this is July. We are now already at almost the end of July, and this is a picture from Uganda, and we see that we haven't got any data from Uganda yet. However, what I want to show now is how this virus is spreading around the continent. This is the number of cases again in Uganda. We see that there's been a disastrous spike and lockdown, and what is associated with it here, B.1.617.2. So basically, when they start sequencing, the virus is associated with a recent massive wave, it's Delta, okay? In the DRC, one of the issues we have is, again, in the DRC, at maximum, we're picking up about 300 cases a day. This isn't one of the largest countries on the African continent. So this is we're just not getting nearly enough testing done. But when they sequence it, and again, these are small numbers, less than 50 samples being sequenced, but still, when they sequence it, my colleagues at INRB, what they find, Delta, okay? I'm using different colors on the different graphs. Sorry, that's a technical limitation, but you can just look at the color here, B.1.617.2, that's Delta again. So again, a big wave, and what is being driven by? Delta, East Africa, Kenya, again, our data is not great recently, but again, we seem to have a growth in cases. And here we've got different variants. The orange is Alpha, and the yellow, which we see growing since May, we don't have much data from June, it's Delta again. So again, a growth in cases is associated with Delta viruses. West Africa, Senegal, another big growth in cases recently. And while it's not very clear what's going on here, they have some other lineages like B.1.416, but we see this purple over here. In June, they are picking up the Delta version of the virus. And I'm pretty certain that once we get July data, we'll find that it is majority Delta again. North Africa, we have no data for Tunisia at the point of this peak. So this is not Delta that is found in May, okay? But you see that we just have this data gap, nothing for June, nothing for July in the public databases, but we see cases are rising precipitously. So that means North, West, Central, Southern, East Africa, we're seeing massive growth, all being driven by this more transmissible version of the Delta virus. And this is in the context of our continent not having nearly enough vaccines. So what's going to happen next with the virus? Because we're talking about Delta, okay? Delta might not be where it ends, but it's very difficult to predict future biology. And because of this, when somebody comes out and maybe their publicity, they're seeking scientists, I'm sorry, but you know, scientists also like showing up on TV sometimes. Beware of scary stories. You know, we heard in India, we heard about double mutant and triple mutants and things like that. We need to study what the impact of the biology is, and it takes time. So you can't just sample one virus and look what the mutations are and say, this is what it's going to do. We don't know enough to know that, okay? But we also don't know that Delta is where it ends. We don't know what other tricks the virus can have in its pockets, okay? And because of this, we need more and we need better eyes. So please, everybody go out there, go lobby the people who pay my salary and tell us that we need more diagnostics. We need better diagnostics, okay? And I'm not only talking about sequencing. The African continent has been left out of the access pool for far too many things, including the testing supplies, right? We should have seen much more before we even got into vaccines. We should have seen much more investment in better diagnostics for the continent. And I mean, honestly, this is not just for this disease, but we are facing diagnostic challenges for all sorts of diseases on the continent, and that is really bad for our populations. Okay. And vaccines. So we know that the vaccines are working against Delta. Okay, we need more and we need to know more. So I said that it hasn't been enough study of some of the effectiveness of the vaccines that we've got to use. You know, we are fixing the car while driving. We have to be doing the studies while doing the rollouts of all our interventions at the moment. Okay, let's have a look at why I say this is a particular concern. This is from Philip Schellekens' website, and he's been arguing that what we're looking at is a shift of the pandemic towards lower income countries. Okay, so let's have a look at this. This is mortality, and in the top, mortality per 100,000 people. Okay, now look at the countries where overall we've seen the worst pandemics. Mostly these are upper middle income countries, high income countries. Okay, of course we know that we've got less good surveillance of deaths in the lower income countries, but still this has been a pandemic that started off as a pandemic that was notable in China, in the USA, in Europe. This is where we saw many deaths. Okay, so in the overall, there's few African countries on this side of the graph: South Africa, Namibia, Tunisia. But now, in the more recent data, the 7-day average of which countries are now being hit worse. Look how many African countries are showing up here: Eswatini, Zimbabwe, Lesotho, Seychelles, Malawi, Namibia, Botswana, South Africa, you know? So we are seeing the pandemic shift from being a pandemic of the global north to being a pandemic of the global south, where Africa is very badly affected. And we're seeing this in terms of the waves. Again, this pink line is a high-income countries, and we see that their deaths recently are way down. okay? Whereas before they had these massive waves. They're very industrialized, very connected countries where air travelers brought in many infections. But now we're seeing that in the middle income and lower income countries, we are starting to see that the deaths are not going down in the way that we saw in the high income countries. And again, looking at this side of the graph, we're seeing a flattening of the curve. And what this really is a picture of, this is a picture of vaccines. These are the guys that have the vaccines and managed to push their deaths down. And these are the guys, these are us, who are waiting for the vaccines or struggling to get the vaccines. Okay, yeah. I'd like to thank my boss, Professor Alan Christoffels, and all of my colleagues at SANBI, and particularly I'd like to also thank some other groups, Bani Jolly, who was part of the group in in India, that identified the rise of Delta, Ino Tool and Verity Hill, that run that Pango lineage system together with many others these days. But the Pango people there at University of Edinburgh and in Andrew and Burroughs' lab, Dr. Emma Hodcroft, who is part of the Next Strain team, and the Next Strain team is what gave all of those trees that I was showing earlier, and then everybody who's been contributing their data to GISAID. It's scary for us to say that we've got a dangerous disease in our continent because we know that we get travel bans, we get all sorts of like negative publicity and things like that. So the ability of African scientists, with the assent of their government, to put the data out there, that has been a courageous move, okay? And it hasn't led to the kind of support that we need on the continent. Okay, and I guess now it's over to you again, Paul.
Yeah, thank you, Peter, for that really, really thorough presentation. And it's quite eye-opening. And I think it answers a lot of questions that many journalists struggle to with the trend of the Delta variant. So before going into any technical question, I have a simple question, which is why is it not possible to really simplify bioinformatics to languages that everybody can understand? Is that remotely possible?
So part of my job is doing exactly that. Historically, the problem is that if you take something like they can have computers that keep your bank running, okay, then that's based on the banking has kind of stayed the same for quite a while, right? If I look at the equipment that I use in bioinformatics, those DNA sequences I was telling you about, the technology is of the last 10, 15 years, okay? So people have come out with new things, and we're basically moving from research. So in other words, people we're doing things as part of their PhD and their very intense study to using it for public health in a very short period of time. So part of my goal is to basically say that, look, I want the public health labs on the continent to be able to run these things without you needing a PhD to understand everything that you're doing. But in my team at SANBI, my software development team, okay, we have like about 4 engineers working on the software. The people who are selling little fluffy toys and cell phones to us at Take-A-Lot, they have 40 people on their team. So none of us is getting sailboat holidays in the Bahamas doing this thing. So there's also a generic underfunding of science and scientific development, which is relating to that no eyes problem that I was telling you about earlier. So yeah. It's a question of basically putting the people and the money into making this thing more accessible, but we are getting there, and especially with the support of Africa CDC, they've been amazing partners, and the WHO, we are going to get there.
So we got a number of questions already for you, and one of the questions that people are interested in knowing is whether bioinformatics and the knowledge that you have can give us any insight into the true origin of the COVID-19 of SARS-CoV-2? Is it remotely possible? And what is the latest on that front?
Yeah, I actually want to move back to this picture. Okay. You can't quite see it here, but one of the things that the sequencing is giving us, and I wish I could zoom into this map, is that if you look very early on, so I talked about these clades, the next train people, they don't use these kind of lots of numbers things. They talk about A clade and B clade. You can imagine it as the big picture as opposed to the Pango lineages, like very detailed picture. Okay. Early on in the outbreak in Wuhan, we saw the emergence of two clades, okay, defined by this L and S difference and a few other differences, okay. Now, what that means to me as a scientist is that if you had somebody leaking a virus from the lab, okay, you wouldn't be seeing two clades like that, okay. They'd be having to leak two viruses that were closely related but different. It's just not what happens. Okay, so what that suggests to me is that there was some, because this virus, it accumulates approximately two changes or two of those RNA changes per month. Okay, so that means that before this, because by the way, those two clades are seen at two different markets in Wuhan. Okay, but what that suggests is that the virus was actually circulating for some time, maybe a month or so, before it got to the markets, which acted as kind of places where a super spreader event happened. Many poor people were infected, okay? Because we know that most people don't, when they get sick, they don't actually transmit the virus to many people, but a small number of people happen to be in the right circumstances to infect a lot of people. So maybe a market vendor or something like that. So all of the evidence we've got at the moment, including the sequence evidence, points to the fact that there was something in animals that were being traded in these markets that allowed them to amplify what was a natural zoonotic infection. And it points away from the idea that this thing either came on frozen food, which is the one theory being pushed out there, or escaped from a lab.
Well, these are really still again propositions, because I think the World Health Organization still described it as inconclusive yet, and would continue to gather evidence. So one question I have, another question we have here is on how these variants arise. We know mutations. Is the Delta variant, did this arise, did this particular mutation arise randomly, or something was intentional that brought about this variant?
So the more that the virus spreads, the more that variants arise, right? So it's a random process of accumulating about 2 changes a month, but you know, it's rolling a dice. Okay, when you're rolling a dice, let's say you're rolling 2 dice. What's the most common thing you're going to get if you roll 2 dice? You're going to get somewhere around 6, you know, but sometimes you get a 12. Okay, and you roll lots of dice and you're going to get those 12s. Okay, so this is a process of random variation, but then feeding into circumstances like, for instance, the mass gatherings that we saw in India, and the quite large outbreak we saw in South Africa and the UK and things like that, that lead to you know, rolling the equivalent of rolling a 12, and then it takes off from there. Okay, but now, you know, if we have a place like a rural area, we are unlikely to see, with low population density, or maybe a lot of young people whose immune system is actually just stopping the virus itself in its tracks. We're unlikely to see a lot of variation happening. But then the other thing is if we have, on the other hand, somebody whose immune system is pushed low, okay, maybe somebody with untreated HIV, then this gives the virus so many chances to like experiment inside their body. So these are some of the factors that lead to new variants arising, but it only needs to hit that 12 and get into the right circumstances for a spread, and then it takes off. And that's what we're seeing with these variants.
Another question that we have is on testing for this particular variant. In your presentation, I think we should take off the screen. Stop sharing the screen. Uh, Yeah, thank you. So your presentation, you mentioned, you brilliantly described why it is really, really difficult to sequence large samples, especially in resource-limited countries. And I'm here wondering, why is it not possible to just test directly for the Delta variant?
It is, but okay, so if you look at like how tests are done, the two main platforms we have are the so-called PCR test and the antigen test. Now the antigen test, that thing is like a little plastic thing, like a pregnancy test. It's manufactured this way. You can't change how it works. So once it's manufactured, it's going to test for the virus in general, and that'll carry on like that. It's the cheaper form of test, but you can't adapt it very much. The PCR test, in theory, you could make a PCR test looking for the specific versions. But the thing is that, again, to reduce the cost of the PCR test, you make the kind of test in bulk, right? In fact, your typical PCR test checks for somewhere on the S protein, somewhere on the E protein, somewhere on the N protein, okay? And to make a new version of the test, now you have to check that its sensitivity is right. You have to roll it out to the regulatory authorities. You have to then distribute it. You see, this is an expensive and slow procedure. So it is possible to do better testing, but it requires investment and interest from people who are test manufacturers. You must understand, on the African continent, we are not really making our own tests to a large extent. We are importing everything.
Yeah, what about elsewhere? Is there any part of the world where they are already testing directly for Delta variant instead of waiting until sequencing is done?
Well, there are, so some people are doing the so-called multiplex PCR where they're testing for different variants of the virus, okay? And I know in Denmark, there's an interesting project where they're using a cheaper form of sequencing, some so-called Sanger sequencing, where they just look at, instead of looking at the whole virus, they just look at that part of that spike protein, and then they rapidly use that to say, well, which variant have we got? So there are people elsewhere in the world who are doing this. But at the moment, the most of the interest has been for clinical reasons, where the question is, are you sick or not? They're not doing it for surveillance regions, which is, where I'm looking at like, well, what exactly are you sick with?
Yeah, so there's also the few, I think yesterday I read the news about Pfizer announcing that a third dose of their vaccine is effective against the Delta variant. Now, is this going to be a new trend with vaccine manufacturers recommending additional doses of their vaccines? Or how do you think the whole interplay of the Delta variant and vaccine?
I think you might have a network problem there, Paul. But I mean, I think that the look, if you're going to walk into a car salesman's room, he's going to try and sell you a car. Okay, I will wait for regulatory authorities to tell us what to do in terms of vaccines, variants, and booster shots before I listen to what the manufacturers of those vaccines are telling me. Yes, we do know that the antibodies fade over time. This is natural. Your body makes antibodies when it sees an infection or you're being injected with a vaccine. And over time, that level of antibodies will naturally fade. This might change, some of how you respond to being infected with a disease, but but if we look at like the data thus far, it's not just the infection we care about, it's a severe disease. And in that regard, the vaccines are still working very, very well, So there doesn't, we shouldn't be like saying it's a good idea to get booster shots just to push your antibodies up. Your antibodies are kind of an artificial measure to some extent. We want to know about the whole immune response and the whole picture of disease that somebody gets. So right now, the regulatory authorities are not agreeing that we're going to need these booster shots. The vaccines do seem to be working. And I think at the moment, we need to suppress the creation of new versions of the virus, which is happening where we have massive outbreaks. So we need to get those vaccines out there to stop these so-called variant factories of these massive outbreaks before we worry about boosting people who've already been vaccinated.
So you think it's not a priority right now?
The priority right now is to deal with where we're seeing these rocket-like spreads that I showed on those graphs. Okay, so your Tunisias, what we saw in the DRC, what we've seen in South Africa, what we've seen in Botswana now recently. The priority is getting the vaccines there, because if we allow the virus to turn Africa into a variant factory, okay, firstly, our economic recovery is going to be impacted, okay, and then secondly, our people are going to die. This is the priority at this point.
And we also have questions about how to protect. What do you think the effective strategy is? I think you mentioned it briefly, and but I think you should still expand on that. So the question was asked specifically for Kenya. How do you think Kenya can deal with the surge in Delta variant?
So one of the problems we've got at the moment is economically and psychologically, our people are quite exhausted. You cannot stay home without somebody effectively paying you to stay at home. Okay, so once the variants are in there, then we had some lockdowns in Uganda and South Africa recently. Yeah, it's possible, but the cost is high. Oh, one thing I will say about the question, they said, look, it looks like it's in Western Kenya at the moment and it's spreading. Yeah, I know we picked up a lot of Delta in Uganda and things like that, but remember there's this time lag in your sequence results. I'm concerned that when people are seeing results, then those results are already old. We mustn't assume that it's only where we are seeing it, okay? Unfortunately, what our experience in South Africa was that once the wave is really kicking off, you can try these blunt measures like lockdowns and things like that. But there's very little that you can do once the wave is already there. We are going to have to just try and push to make sure that the next wave doesn't happen.
Now, the question that another question we have here is, what do you think journalists are getting wrong in their Delta variant reporting, and what do you think they are not paying attention to?
So the one thing that worried me a few months ago is when as I said, from December, it became talking about the variants. Okay, everybody was talking about everybody had a variant. Nigeria had a variant. There's South Africa had some variants, and everybody wanted to talk about a new variant. Okay, so we need to keep a focus on the fact that this is actually all one pandemic, and not get too distracted by what I call scariant stories, where it's only about the scary variants. And I know that people are kind of sick and tired of talking about COVID-19 already, but I felt like people were sometimes just looking for the kind of clickbait stories about this particular worrying variant and not paying attention to the maybe boring, ordinary, existing stuff that's already happening. The virus has largely been causing the same sickness. That was my one concern. And the other thing is numbers, people. Okay, we have a lot of attention being paid to people who got vaccinated and then got sick. Okay, these so-called breakthrough infections, and then it's important to realize that no vaccine is 100% effective. The really important thing is what is the spectrum of illness that we're getting? So are they getting sick, or are they ending up in hospital and dying? So we have to look beyond those basic case numbers, because if we look in the UK at the moment, people are getting sick, but they're not dying to the same degree that we saw before, okay? And that distinction is important. And the last one is that when we are seeing quite a bit of irresponsible reporting in terms of this one got a vaccine and then they died, okay? Look, unfortunately, people die on a regular basis. The question is there something that's associated with a vaccine? Or is it just happening and it happens in approximately the same time as the vaccine. I mean, I can get vaccinated today, and tomorrow I can be in a car crash and die. Okay, the vaccine is not stopping death, but we have to put things within a numerical context. So I've seen some very bad mistakes in terms of not looking at the bigger picture of what's going on and only looking for the thing that's going to make a juicy headline.
Juicy headline. That really hits right deep in the heart of what some journalists are actually doing. And if we look at, even though we are talking about the Delta variant, do you think there are other variants that that you are keeping track of and should be of interest to everybody?
Well, look, there's now talk of a Lambda variant. So long as the pandemic continues, there will be changes. There will be more variants, and unfortunately, often we'll pick them up too slowly and too late. So yeah, so we need to keep an eye on this, and we need to keep an eye on the continuing studies on the best interventions against the virus. And but mostly I think that scientists and journalists need to work together, because actually by now, by July 2021, we all have meant to have become a little bit of a virologist, a little bit of a scientist. You know, our populations are meant to have been educated during this pandemic. And this is something where both as scientists and I believe as journalists and as members of the public in general, we all need to work harder, because there are people out there making money off misinformation, and we all have a fight together against them.
Now, if you look at from what we are getting from the trend, the emergence, and the rapid spread of these variants, what do you think these infer about how long the pandemic will still be around? And is there any green lights at the end of the tunnel?
Look, we are not on a rich continent. Unfortunately, we are in this situation where how long will the pandemic last? We have part of the answer, obviously, in our behavior and what our governments are doing and things like that. But a big part of the answer is in the people with the resources to increase the rollout of vaccines, diagnostics, and other interventions, okay? So unfortunately, we kind of have, it's kind of a political question. How long will humans allow the pandemic to carry on? And it's not really us humans here, the kind of ordinary things like that, but you know, the CEOs of big companies and the leaders of rich nations. So how long will they allow the pandemic to carry on? Okay, I mean, this is what Dr. Tedros from the WHO and Dr. Ryan Kerkhove have been saying. This is in whose hands the pandemic is. And I mean, unfortunately, it feels like this is a taste for the future to come. You know, when there's a crisis and it only affects us here on the African continent, it seems that the crisis is allowed to carry on forever. I'm hoping that the political pressure, both within those high income countries and from some of our leaders here, can force those who hold the wealth of the world to realize that the time to end the pandemic is now.
Do you think we have all it takes to end the pandemic across the world right now?
Yeah, we have it. It's just unevenly distributed. We have the vaccines. Look, the estimate is that the cost of scaling up vaccines to be able to get every single human vaccinated before the end of this year is less than 30 billion US dollars. Now that sounds like a lot of money until you realize how much the budgets of high income countries is. So for less than 30 billion US dollars, we could be scaling up vaccine production and getting vaccines into everybody's arms before the end of this year.
I was about to wrap up when these questions snuck in, and it goes thus. When it comes to knowledge translation, where do you see areas that could improve, excluding journalists? How and what recommendations can other disciplines help in making us more knowledgeable?
Well, the one thing that I feel has been underutilized in this pandemic is the social scientists, okay? Those who can help, because I just talk, I'm a scientist, I'm a geek and things like that. There are those who study how people hear and how we can tune the message. The other thing is we cannot stay in English, you know. In my country, South Africa, majority of people, their home language is not English. And I think this is true in most of our countries on the continent. Even between the Francophone and the Anglophone and so on, and the Arabic speaking, we've had some communication issues, you know. But it's vital that we figure out how to do knowledge translation into African languages, not just the ones that we inherited from colonialism.
Then what would you describe to be your passing words? What are last remarks for our journalists who are in attendance today?
Well, thanks for listening. And I mean, I'd like to salute all of the African scientists that have been working on this pandemic. And in a sense, yeah, we are not all in this together, but we can all play a part if we can like embrace each other more closely than we do. So the biggest enemy we have at the moment is division, and those who profit from division. So I think that the future that we need to find is unity, and yeah, I look forward to that in the future.
So thank you very much for being for taking our time to be with us today. I really appreciate you for accepting our invitation and for all the insights that you've shared with journalists in different parts of the world. Like you said, it's something that is not just combating this pandemic is not a responsibility of one person. It's a collective effort from the scientists to the geeks like you, to the social scientists, to those that are making the policies and are in charge of influencing real changes. This is all encompassing. And I hope journalists on this call today, they've been able to expand their knowledge on Delta reporting. And like he rightly said, it's not just about the single variant. It's a whole ecosystem of the virus itself that continues to evolve because it is spreading from one person to another. And as the spread continues, as spread rapidly continues, we expect to still see the virus. We getting those lucky number 12, lucky 12 that would have different unique features like the Delta. The next strain variant could be even more severe than the Delta. It's all about breaking the transmission, and breaking the transmission requires getting access to vaccines and dealing with the fatigue that many people across the world are dealing with. So thank you today for joining us on this webinar. And if you need for more information about the ICFJ Global Health Crisis Reporting Forum, please visit www.icfj.org. And for more information on to access more resources for journalists, we have lots of resources that you can utilize in many aspects, in any area, on any issue that affects you as a journalist. Please visit www.ijnet.org. Next week, we'll be back again with another interesting and exciting webinar with another resource person who will also be talking about how everyone has become a health reporter today, sharing a personal experience of a journalist that went from just reporting politics to reporting COVID. And it's going to be an eye-opener on how to start health reporting. So from myself and everybody at the ICFJ, I say thank you and have a great day. Bye. Bye, Peter.